Hello everyone. Thank you for viewing my poster. My name is Sharel Cornelius and I'm a PhD student at the Department of Chemistry and Biochemistry at the University of Texas at Arlington. I will be discussing my research through this poster, which is titled as The Study of Scrambling Effect of Disulfides in Proteins and Peptides. The cysteine residues which are present in the proteins and peptides have thiols as their side chains, which tend to form disulfide bonds either by the process of thiol exchange or by the oxidation of cysteine residues. The disulfide bonds so formed play a significant role in maintaining the structure and function of the proteins, and the disulfide bonds so formed can be of different types. They can either be interchain, means they are forming within two different peptides. Intrachain means they are forming within the same peptides, or a combination of both. The misorientation or the scrambling of disulfide bonds affects the structure and function of proteins, and a few common neurodegenerative misfolding diseases like Alzheimer's and Huntington's disease have been found to occur as a result of formation of aggregates in proteins due to the disulfide bond scrambling. The disulfide bond scrambling also affects the functionality of antibodies as they are exposed to various pH changes during their manufacturing, which further leads to the scrambling of disulfide bonds in solutions. A very common method for the study of disulfide bonds includes the profile comparison method. And our study of disulfide bonds focuses on the comparative analysis of reduced and non-reduced disulfide bonded intact proteins using mass spectrometry, and the experimental approach facilitates the chemical modification of disulfide bonds in proteins using melamine-based reagent. Our workflow for the study of disulfide bond proteins includes the tryptic digestion of proteins in solution without any reduction and alkylation of the disulfide bonds, and then we compare the results with a tryptic digest of the same protein along with the reduction and alkylation of the disulfide bonds. So considering a standard protein called BSA, I was able to probe the native disulfide bonds and the scrambled disulfide bonds in the protein, and in figure 5 you can see the red lines, the red connective lines represent the scrambling that happened in the protein, and the dotted green lines represent the native linkages in the BSA protein. To further probe and characterize and also enrich the disulfide bonds from complex mixture of peptides and proteins, our lab developed a melamine-based reagent with dual functionality. So the reagent is capable of binding to the disulfide bonds through bioconjugation, and also it can bear an enrichment tag which can then be useful for the enrichment of disulfide bonds from complex mixture of proteins. The chemical modification of the disulfide bonds in lysozyme protein was successfully attained by labeling the disulfide bonds with our melamine-based reagent after the reduction of the disulfide bonds with TSAP, which is a reducing agent which does not have any thiols in it. Figure 7 and figure 8 are the tandem mass spectrometry results for the bioconjugation and the chemical labeling of the disulfide bonds in lysozyme protein. So, in conclusion, a quick reduction and labeling of disulfide bonds with melamine-based reagent leads to the development of an excellent method for their identification and characterization using collision-induced dissociation mass spectrometry, along with the reduction of the disulfide scrambling that happens in solutions. And also the melamine-based reagent can be utilized for the selective enrichment and identification of disulfide linkages in intact proteins and complex systems. Thank you.